even though the research behind it is absolutely remarkable. Wild salmon. I know what some of you are thinking. Fish is good for the brain. I have heard that 1,000 times. Tell me something new. But hold on, because what I am about to explain goes far beyond the generic advice to eat more fish. The specific reason wild salmon transforms your brain during sleep is a mechanism that most health content completely ignores, and it has to do with something called the phospholipid bilayer. Every single one of your 100 billion brain cells is surrounded by a membrane. That membrane is not just a wall. It is a living, active structure that controls everything entering and leaving the cell. It controls how signals pass between neurons. It controls how efficiently your brain cells communicate with each other. And that membrane is made primarily of a specific type of fat called docosahexaenoic acid. You might know it better as DHA. DHA is not just one of many fats in your brain. It is the dominant structural fat in your cerebral cortex, the outer layer of your brain responsible for thinking, reasoning, and memory. It makes up somewhere between 15 and 20% of the total fat content of your brain. And here is where the sleep connection comes in. During deep sleep, when your brain is consolidating memories and strengthening synapses, the integrity of those cell membranes determines how cleanly and efficiently signals can travel between neurons. If your cell membranes are stiff, if they are degraded, if they are made of the wrong types of fats, then signal transmission becomes noisy and inefficient. It is like trying to have a phone conversation on a bad connection. The information gets garbled. Memories do not form properly. Consolidation fails. After 60, the DHA content of your brain naturally declines. This has been measured in autopsy studies. Older brains consistently show lower concentrations of DHA in their cell membranes compared to younger brains. And this decline directly correlates with measurable reductions in cognitive function and memory performance. Now here is why wild salmon specifically. Wild salmon, particularly sockeye salmon, contain some of the highest concentrations of DHA found in any food on the planet. A single 6-ounce serving of wild sockeye salmon delivers roughly 1,500 milligrams of DHA. That is a massive dose. And unlike plant-based omega-3 sources like flaxseed or walnuts, which contain a precursor called ALA that your body must convert into DHA through an extremely inefficient process, the DHA in salmon is preformed. It is ready to go. Your body can absorb it and shuttle it directly to your brain without any conversion needed. But DHA is not the only reason salmon earns the top spot on this list. Wild salmon is also one of the richest dietary sources of a compound called astaxanthin. Astaxanthin is what gives salmon its distinctive pink-red color. It is a carotenoid pigment produced by the microalgae that salmon eat throughout their lives. And astaxanthin has a property that makes it almost uniquely valuable for brain health. Most antioxidants cannot cross the blood-brain barrier. This is the tightly regulated gateway that controls which molecules can enter your brain from your bloodstream. It is an essential protective mechanism, but it also means that many of the antioxidants you consume in food never actually reach your brain tissue. They protect your heart, your liver, your muscles, but your brain does not benefit from them. Astaxanthin is one of the rare exceptions. It crosses the blood-brain barrier efficiently. Once inside your brain, it embeds itself directly into cell membranes, where it provides antioxidant protection exactly where it is needed most. It neutralizes free radicals right at the membrane surface, preventing the lipid peroxidation that degrades DHA and damages cell membranes over time. Think about what this means for your sleeping brain. During the night, your brain is running high-energy repair operations. These operations produce oxidative stress as a byproduct. Free radicals are generated, and if those free radicals are not neutralized, they damage the very cell membranes your brain is trying to repair. Astaxanthin acts as an on-site bodyguard, protecting the repair work as it happens. A study published in the British Journal of Nutrition found that astaxanthin supplementation in older adults improved cognitive function and reduced markers of oxidative stress in the brain. Another study, published in the journal Marine Drugs, demonstrated that astaxanthin reduced the accumulation of beta amyloid plaques in animal models, directly supporting the glymphatic cleaning process I described earlier. And there is one more piece to the salmon story that I want to highlight. Wild salmon is an excellent source of vitamin B12. After 60, B12 absorption declines significantly due to age-related reductions in stomach acid production. And B12 deficiency is directly linked to brain shrinkage. Not metaphorically, literally. Studies using brain imaging have shown that people with low B12 levels have measurably smaller brain volumes and faster rates of brain atrophy compared to people with adequate levels. B12 is essential for maintaining the myelin sheath, the insulating layer that wraps around nerve fibers and allows electrical signals to travel quickly and efficiently between neurons. When myelin degrades, signal transmission slows down. Thinking becomes sluggish. Reaction times increase. Memory retrieval takes longer. By providing a highly bioavailable source of B12, wild salmon helps maintain the structural integrity of the wiring that your brain depends on during its nighttime consolidation work. So let me summarize why wild salmon is food number one. It delivers preformed DHA to rebuild and maintain brain cell membranes. It provides astaxanthin that crosses the blood-brain barrier to protect those membranes from oxidative damage during sleep, and it supplies B12 to maintain the myelin insulation that keeps neural signals fast and clean. 3 separate mechanisms, all from a single food, all directly supporting what your brain does while you sleep. Now I want to emphasize one word in the name of this food: wild. Wild salmon is fundamentally different from farmed salmon in its nutritional profile. Farmed salmon has significantly lower levels of astaxanthin because farmed fish are often fed synthetic versions of the pigment rather than accumulating natural astaxanthin from wild algae. Farmed salmon also tends to have a less favorable omega-3 to omega-6 ratio, higher levels of environmental contaminants, and lower overall nutrient density. If you can get wild-caught salmon, particularly sockeye or king salmon, that is the gold standard. 2 to 3 servings per week is a solid target, and if you eat one of those servings in the evening, within 3 to 4 hours of sleep, you are providing your brain with the building blocks it needs right when the repair shift begins. Let us move on to food number 2, and this one is going to take you somewhere you probably did not expect. Tart cherries. If salmon is the construction crew that rebuilds your brain at night, tart cherries are the shift manager that makes sure the crew actually shows up on time. Because tart cherries do something that no other food on this list does. They directly regulate the timing and depth of the sleep itself, the very process that makes all of this nighttime repair possible. Tart cherries, specifically the Montmorency variety, are one of the only significant natural food sources of melatonin. Not a supplement. Not a synthetic version. Actual food-derived melatonin. And before you say, wait, I thought melatonin was just a sleep supplement. Let me explain why melatonin is far more important to your brain than you probably realize. Melatonin is a hormone produced by your pineal gland in response to darkness. Its primary role is to regulate your circadian rhythm, the internal clock that tells your body when to sleep and when to wake. But melatonin is also a potent antioxidant, and not just any antioxidant. Melatonin has a special property called cascade antioxidant activity. When most antioxidants neutralize a free radical, they become inactive afterward. One and done. Melatonin, on the other hand, generates metabolites when it neutralizes a free radical, and those metabolites are themselves antioxidants. So a single molecule of melatonin can neutralize multiple free radicals in a chain reaction. This makes it extraordinarily efficient at reducing oxidative damage in the brain. Now, here is why this matters so much after 60. Melatonin production declines dramatically with age. A 70-year-old produces roughly half the melatonin of a 25-year-old, sometimes even less. This decline has 2 devastating consequences for brain health. First, it disrupts deep sleep architecture. You spend less time in the slow-wave deep sleep stages where glymphatic cleaning and memory consolidation occur. You wake up more frequently during the night. The total amount of restorative sleep you get shrinks even if you are in bed for the same number of hours. Second, you lose the antioxidant protection that melatonin provides to your brain during those critical overnight hours. This is where tart cherries come in. Research from Louisiana State University found that drinking tart cherry juice twice daily increased sleep time by an average of 84 minutes in older adults with insomnia. 84 minutes. That is almost an hour and a half of additional sleep, including additional time in the deep sleep stages where brain repair occurs. But the benefits go beyond just the melatonin content. Tart cherries contain a class of compounds called anthocyanins. These are the deep red and purple pigments that give the cherries their color, and anthocyanins are among the most powerful anti-neuroinflammatory compounds found in any food. Let me explain why neuroinflammation is such a big deal for your sleeping brain. Inflammation in the brain is not like inflammation in your knee or your shoulder. You cannot feel it. There is no swelling, no redness, no pain. Brain inflammation is driven by cells called microglia, which are the immune cells of the central nervous system. When microglia become chronically activated, which happens increasingly with age, they release inflammatory molecules called cytokines that damage nearby neurons and synapses. This chronic low-grade neuroinflammation is one of the primary drivers of age-related cognitive decline. It interferes with synaptic plasticity, the ability of synapses to strengthen and reorganize during sleep. It impairs memory consolidation. It increases the production of beta amyloid, and it creates a toxic environment that makes it harder for your brain to repair itself, even when you do get adequate deep sleep. The anthocyanins in tart cherries have been shown in multiple studies to reduce neuroinflammation by inhibiting the activation of a protein complex called nuclear factor kappa B, often written as NFKB. This is one of the master switches that controls inflammatory gene expression throughout the body, including in the brain. When anthocyanins suppress NFKB activation, they dial down the production of inflammatory cytokines, giving your brain a cleaner, calmer environment to perform its nighttime repair work. A study published in the journal Food and Function found that older adults who consumed tart cherry juice daily for 12 weeks showed significant improvements in memory, particularly in tasks related to visual and spatial memory. Another study, published in Nutrients, found that tart cherry consumption reduced markers of oxidative stress and inflammation in the brains of aging animal models. And here is a detail that I find particularly fascinating. Tart cherries also contain meaningful amounts of tryptophan, the amino acid precursor to serotonin, which in turn is the precursor to melatonin. So tart cherries are not just delivering melatonin directly. They are also providing the raw material your brain needs to manufacture more melatonin on its own. It is both an immediate supply and a long-term production boost. Now the form matters. When I say tart cherries, I mean genuinely tart varieties like Montmorency. Sweet cherries, the kind you buy in the produce section to snack on, do not contain the same concentrations of melatonin or anthocyanins. You want either frozen tart cherries, dried tart cherries with no added sugar, or 100% tart cherry juice concentrate. The ideal timing is about 1 to 2 hours before bed. This aligns the melatonin boost with your natural pre-sleep window. And combining tart cherries with one of the other foods on this list, I will explain how at the end creates a synergistic effect that amplifies the brain repair process significantly. But before we get there, we need to talk about food number 3. And this one might be the most underrated brain food in existence. It has been sitting in grocery stores for decades, it costs almost nothing, and the research supporting its effects on brain repair during sleep is staggeringly strong. Food number 3, eggs. Not egg whites, whole eggs, specifically the yolk, because the yolk of an egg contains what I consider the single most important nutrient for nighttime brain repair that almost nobody is talking about. That nutrient is choline. Choline is not a vitamin. It is not a mineral. It is an essential nutrient that sits in its own category, and your brain is absolutely desperate for it. Choline is the primary building block of acetylcholine, one of the most critical neurotransmitters in your entire nervous system. Acetylcholine is the neurotransmitter most directly involved in memory formation, learning, attention, and the regulation of sleep architecture, particularly the REM sleep stage where emotional memory processing and creative problem solving occur. After 60, acetylcholine levels decline significantly. This decline is so central to age-related cognitive impairment that the primary class of drugs used to treat Alzheimer's disease, called cholinesterase inhibitors, works by preventing the breakdown of whatever acetylcholine remains. These drugs do not create new acetylcholine. They just try to keep the existing supply from being destroyed too quickly. That is how important this single neurotransmitter is to brain function. And here's the problem. The vast majority of people over 60 are not consuming anywhere near enough choline. A study published in the Journal of the American College of Nutrition found that less than 10% of the American population meets the adequate intake for choline. Less than 10%. That means over 90% of people are chronically undersupplying their brains with the very nutrient it needs most for memory and overnight repair. One large egg yolk contains approximately 150 milligrams of choline. The adequate daily intake for men over 60 is 550 milligrams. For women over 60, it is 425 milligrams. 2 to 3 whole eggs per day put you well on your way to meeting that target. And egg yolk choline is in a form called phosphatidylcholine, which is highly bioavailable and can be readily incorporated into brain cell membranes and used for acetylcholine synthesis. But choline is not the only reason eggs belong on this list. Egg yolks are one of the best dietary sources of lutein and zeaxanthin, two carotenoid pigments that most people associate with eye health, but that have increasingly been linked to brain health as well. Research from the University of Illinois found that lutein accumulates in brain tissue, and that higher brain lutein concentrations are associated with better cognitive performance in older adults. A study published in the journal Neurobiology of Aging found that adults with higher blood levels of lutein and zeaxanthin had more efficient neural processing and better memory performance. The mechanism appears to involve both antioxidant protection and anti-inflammatory activity within the brain, particularly in regions associated with memory and learning. And there is more. Egg yolks contain meaningful amounts of vitamin D. This is relevant because vitamin D deficiency is extremely common in adults over 60, particularly in northern latitudes and in people who spend most of their time indoors. And vitamin D is not just a bone nutrient. It is a neurosteroid that plays active roles in brain function. Vitamin D receptors are found throughout the brain, with particularly high concentrations in the hippocampus, the very region responsible for memory formation and the primary site where memory consolidation occurs during sleep. Research published in the journal Neurology found that older adults with severe vitamin D deficiency had more than double the risk of developing dementia compared to those with adequate levels. Another study published in JAMA Neurology found that low vitamin D status was associated with faster rates of cognitive decline over a multi-year follow-up period. Eggs also provide complete protein with all essential amino acids, including tryptophan, which, as I mentioned with tart cherries, is the precursor to serotonin and ultimately melatonin, and they provide vitamin B12 and folate, both of which are involved in homocysteine metabolism. Elevated homocysteine is a well-established risk factor for brain atrophy and cognitive decline, and adequate B12 and folate intake keeps homocysteine levels in check. Now I want to address the elephant in the room because I know some of you are thinking it. What about cholesterol? For decades, eggs were demonized because of their cholesterol content. People were told to limit eggs, avoid yolks, eat only the whites. And many people over 60 still follow that outdated advice because it was drilled into them for so long. Here's what the current science actually says. The dietary cholesterol in eggs has minimal impact on blood cholesterol levels in the vast majority of people. Your liver produces the majority of the cholesterol in your blood, and when you consume more cholesterol from food, your liver simply produces less to compensate. This regulatory mechanism has been confirmed in multiple large-scale studies. The 2015 to 2020 dietary guidelines for Americans removed the previous recommendation to limit dietary cholesterol to 300 milligrams per day, specifically because the evidence did not support it. More importantly, cholesterol is actually essential for brain function. Your brain contains roughly 25% of all the cholesterol in your body, despite being only about 2% of your body weight. Cholesterol is a critical structural component of brain cell membranes and is essential for synapse formation and maintenance. Depriving your brain of cholesterol-rich foods while your brain is trying to rebuild synapses during sleep is counterproductive. So to summarize food number 3, whole eggs provide choline for acetylcholine production and memory consolidation. They provide lutein and zeaxanthin for neuroprotection. They provide vitamin D for hippocampal function. They provide tryptophan for melatonin production. They provide B12 and folate for homocysteine regulation. And they provide cholesterol as a structural building block for brain cell membranes. All of this from one of the cheapest, most accessible, most versatile foods on the planet. 2 to 3 eggs daily with the yolks is a reasonable and well-supported target for brain health. If you have been throwing away yolks on the advice of outdated dietary guidelines, it is time to reconsider that habit. Your sleeping brain needs what those yolks contain. Now let us talk about food number 4. And this is the one that, when I explain the mechanism, is going to fundamentally change how you think about a food you probably already have in your kitchen. Walnuts. Every food on this list so far has targeted a specific aspect of nighttime brain repair. Salmon rebuilds cell membranes. Tart cherries optimize the sleep that makes repair possible. Eggs fuel neurotransmitter production and provide structural building blocks. But walnuts do something different. Walnuts target the repair process itself at the genetic level. They influence which genes in your brain cells get turned on and which get turned off during sleep. And that is an extraordinary thing for a food to do. The primary mechanism here involves a compound found in walnuts called ellagic acid. When you eat walnuts, the ellagic acid is metabolized by your gut bacteria into compounds called urolithins, particularly urolithin A. Urolithin A has been one of the hottest topics in aging research over the past several years, and for good reason. It activates a cellular process called mitophagy. Mitophagy is your cell's quality control system for mitochondria. Mitochondria, as you may recall from biology class, are the energy-producing structures inside every cell. Your brain cells are among the most energy-hungry cells in your body. Despite making up only 2% of your body mass, your brain consumes roughly 20% of your total energy, and that energy comes from mitochondria. As you age, mitochondria accumulate damage. Their internal DNA, which is separate from the DNA in your cell nucleus, is particularly vulnerable to oxidative damage because it lacks the protective histone proteins that shield nuclear DNA. Damaged mitochondria become less efficient at producing energy and start generating more free radicals as a byproduct. They become like old, malfunctioning factories that produce less product and more pollution. Mitophagy is the process by which your cells identify these damaged mitochondria, break them down, and recycle their components to build new, healthy mitochondria. It is the ultimate cellular renewal process, and it happens primarily during sleep, when your cells shift from performance mode to maintenance mode. After 60, mitophagy becomes less efficient. Damaged mitochondria accumulate. Brain cells progressively lose energy production capacity, and this energy deficit directly impairs every aspect of nighttime brain repair. The glymphatic system requires energy to drive fluid flow. Memory consolidation requires energy for synaptic strengthening. Neurotransmitter production requires energy. Without adequate cellular energy, the entire repair system slows down. Urolithin A from walnut consumption has been shown to restore mitophagy efficiency. A landmark study published in Nature Medicine demonstrated that urolithin A improved mitochondrial health and muscle function in older adults. Subsequent research has shown similar benefits in brain tissue, with improved mitochondrial function in neurons leading to better energy production and reduced oxidative stress. But urolithin A is not the only brain-relevant compound in walnuts. Walnuts are also the richest tree nut source of alpha-linolenic acid, the plant-based omega-3 fatty acid. While I mentioned earlier that ALA conversion to DHA is inefficient, ALA has its own independent anti-inflammatory effects in the brain. It inhibits the production of pro-inflammatory eicosanoids and helps maintain the fluidity of cell membranes in ways that complement the DHA from salmon. Walnuts also contain exceptionally high levels of polyphenolic compounds. A study published in the Journal of Nutrition found that walnuts have the highest total polyphenol content of any commonly consumed nut, nearly twice the amount found in almonds, peanuts, or pistachios. These polyphenols, including pedunculagon, telemigrandin, and casuarictin, have demonstrated potent antioxidant and anti-neuroinflammatory effects in laboratory studies. And here is a particularly compelling finding. The Nurses' Health Study, one of the largest and longest-running nutritional epidemiological studies ever conducted, followed over 19,000 women over a period of years, and found that regular walnut consumption was associated with significantly better cognitive function in later life. Women who consumed 5 or more servings of nuts per week, with walnuts being the most frequently consumed nut, performed measurably better on cognitive tests compared to non-nut consumers. Another study, published in the Journal of Alzheimer's Disease, found that supplementing the diet with walnuts improved learning ability and memory in animal models and significantly reduced beta amyloid accumulation. The researchers attributed these effects to a combination of the antioxidant, anti-inflammatory, and mitophagy-enhancing properties of walnut compounds. There is also a fascinating structural detail about walnuts that I cannot help mentioning, even though it is not scientific evidence. Have you ever looked at a walnut closely? When you crack open the shell, the walnut inside is shaped remarkably like a brain. 2 hemispheres, wrinkled folds that resemble the gyri and sulci of the cerebral cortex. It is one of those interesting coincidences that has caught people's attention for centuries. The doctrine of signatures, an old philosophical concept, suggested that foods resembling body parts would benefit those organs. In the case of walnuts and the brain, the modern research has actually confirmed what ancient observers speculated. Sometimes pattern recognition gets it right. The ideal amount is roughly 1 ounce per day, which is approximately 14 walnut halves or a small handful. This amount consistently shows up in the research as the threshold where cognitive benefits become measurable. Eating them in the evening allows the ellagic acid to be metabolized by your gut bacteria overnight, producing urolithins right when your cells are entering their maintenance and repair phase. One additional note on walnuts. The gut bacteria that convert ellagic acid into urolithin A are not present in everyone's microbiome at the same levels. Regular walnut consumption actually helps cultivate these beneficial bacteria over time, meaning the benefits tend to increase the longer you maintain the habit. This is a food where consistency matters even more than quantity. Now we have covered 4 foods, each targeting a different pillar of nighttime brain repair: membrane integrity, sleep optimization, neurotransmitter production, cellular energy renewal. The 5th and final food targets what might be the most critical pillar of all, and it is the one most people over 60 are getting absolutely wrong. Food number 5, spinach, specifically dark leafy spinach. Not iceberg lettuce, not the pale baby spinach that comes pre-washed in plastic containers, although that is better than nothing. I am talking about mature, dark green, deeply pigmented spinach leaves. And the reason this food completes our list comes down to one word that you may not have heard before in the context of brain health: methylation. Methylation is one of the most fundamental biochemical processes in your body. It involves the transfer of a single carbon atom bonded to 3 hydrogen atoms, called a methyl group, from one molecule to another. This sounds abstract, but methylation controls an astonishing range of biological functions. It controls gene expression. It regulates neurotransmitter production and breakdown. It maintains DNA integrity. It governs the building and repair of myelin. It regulates the inflammatory response. It is involved in hundreds, possibly thousands, of biochemical reactions that keep your brain functioning properly. During sleep, methylation activity increases significantly. Your brain uses methylation to repair DNA damage accumulated during the day. It uses methylation to regulate the expression of genes involved in synaptic plasticity and memory consolidation. It uses methylation to produce and recycle the neurotransmitters that facilitate nighttime brain processes. Methylation is, in many ways, the invisible operating system running beneath all the other repair mechanisms we have discussed. And methylation depends critically on a single nutrient: folate, natural folate, not folic acid, which is the synthetic form found in supplements and fortified foods. Natural folate, the kind found in dark leafy greens like spinach. Your body handles natural folate and synthetic folic acid very differently. Natural folate from food is converted in the gut into a form called 5-methyl tetrahydrofolate, which enters your bloodstream and is ready for immediate use. Synthetic folic acid, on the other hand, must be converted by your liver through a multi-step enzymatic process, and a significant percentage of the population has genetic variants, particularly in the MTHFR gene, that make this conversion slow and inefficient. Some estimates suggest that up to 40% of the population has some degree of reduced MTHFR function. For these people, supplemental folic acid is a poor substitute for food-based folate. Spinach is one of the most concentrated dietary sources of natural folate on the planet. A single cup of cooked spinach provides approximately 260 micrograms of folate, which is over 60% of the daily recommended intake. And this food-based folate is in the form your body can actually use efficiently, regardless of your MTHFR genetic status. But spinach is not just a folate delivery system. It is a comprehensive brain repair food that attacks cognitive decline from multiple angles simultaneously. Spinach is extremely rich in nitrates. Dietary nitrates are converted by bacteria in your mouth and gut into nitric oxide, a molecule that dilates blood vessels and improves blood flow. And blood flow to the brain is not a trivial issue for nighttime repair. Every nutrient, every molecule of oxygen, every building block that reaches your brain cells during sleep gets there through blood vessels. If blood flow is restricted, which it increasingly is after 60, due to arterial stiffness and endothelial dysfunction, then the delivery system for nighttime repair is compromised. A study from Wake Forest University found that a high nitrate diet increased blood flow specifically to the frontal lobes of the brain in older adults. The frontal lobes are responsible for executive function, planning, decision-making, and working memory, all functions that decline with age. Another study, published in the American Journal of Clinical Nutrition, found that dietary nitrate supplementation improved cognitive performance in older adults, particularly in tasks requiring sustained attention and processing speed. The timing here is significant. Nitric oxide production from dietary nitrates follows a slow, sustained release pattern. Eating spinach with dinner means that nitric oxide levels in your blood will be elevated throughout the early and middle portions of your sleep, precisely when deep, slow-wave sleep and the associated brain repair processes are most active. You are essentially opening the highways that deliver repair materials to your brain right when the repair crew needs them most. Spinach also contains exceptionally high levels of magnesium. 1 cup of cooked spinach provides roughly 157 milligrams of magnesium, making it one of the richest food sources available. Magnesium is directly involved in over 600 enzymatic reactions in your body, and its role in brain function is multifaceted. Magnesium regulates NMDA receptors, which are the primary receptors involved in synaptic plasticity and long-term potentiation, the molecular mechanism of memory formation. Research from the Massachusetts Institute of Technology found that a specific form of magnesium increased synaptic density and improved both short-term and long-term memory in aging animal models. The researchers found that elevated brain magnesium levels enhanced the brain's ability to strengthen synaptic connections, the very process that memory consolidation during sleep depends on. Magnesium also promotes deep sleep. It activates the parasympathetic nervous system, the rest and digest mode that prepares your body for sleep. It regulates GABA receptors, the primary inhibitory neurotransmitter system that calms neural activity and facilitates sleep onset. And it helps regulate cortisol, the stress hormone that, when elevated at night, disrupts sleep architecture and impairs nighttime brain repair. Spinach is also one of the richest dietary sources of vitamin K1. Now, vitamin K is typically associated with blood clotting and bone health. But emerging research has revealed a critical role for vitamin K in brain health that was previously unknown. Vitamin K is involved in the synthesis of sphingolipids, a class of fats that are essential components of brain cell membranes. Sphingolipids are particularly concentrated in myelin, the insulating sheath around nerve fibers. A study published in the journal Neurobiology of Aging found that higher dietary vitamin K intake was associated with better episodic memory in older adults. Another study found that vitamin K status was a significant predictor of cognitive performance in elderly populations, independent of other nutritional factors. By providing abundant vitamin K, spinach supports the sphingolipid synthesis that maintains myelin integrity and facilitates the fast, efficient neural communication your brain depends on during memory consolidation. Finally, spinach provides substantial amounts of vitamin E, particularly alpha-tocopherol, which is the form of vitamin E most efficiently retained by the brain. Vitamin E is a fat-soluble antioxidant that embeds itself in cell membranes and protects them from lipid peroxidation, working alongside the astaxanthin from salmon to form a comprehensive membrane defense system. So let me bring this all together. Spinach provides natural folate to fuel the methylation reactions that underlie DNA repair, gene expression, and neurotransmitter metabolism during sleep. It provides nitrates to increase blood flow to the brain, ensuring adequate delivery of oxygen and nutrients during nighttime repair. It provides magnesium to enhance synaptic plasticity, promote deep sleep, and support the molecular machinery of memory consolidation. It provides vitamin K to maintain sphingolipid and myelin synthesis, and it provides vitamin E to protect brain cell membranes from oxidative damage. 5 distinct mechanisms from a single food. 2 cups of fresh spinach or 1 cup of cooked spinach is a reasonable daily target. Cooking spinach actually increases the bioavailability of several of its key nutrients, including folate and lutein, because it breaks down the cell walls that would otherwise limit absorption. A warm spinach sauté with dinner, seasoned with olive oil to enhance absorption of the fat-soluble vitamins, is an easy and effective way to deliver these brain-critical nutrients in the hours before sleep. Now that you understand each food individually, I want to pull back and show you the bigger picture, because these 5 foods were not chosen randomly. Each one was selected because it fills a specific gap in your brain's nighttime repair process that the other 4 do not. Together, they form a complete system. Think of it this way. Imagine your brain's overnight repair operation as a construction site. Wild salmon provides the building materials, the DHA for cell membranes, the B12 for myelin, the astaxanthin for on-site damage protection. Tart cherries are the scheduling manager, ensuring the work actually happens by optimizing the depth and duration of the sleep that makes repair possible, while also suppressing the inflammation that would otherwise interfere with the construction. Eggs provide the fuel and the chemicals, the choline for acetylcholine production, the vitamin D for hippocampal signaling, the cholesterol for membrane construction. Walnuts are the quality control team, triggering mitophagy to clear out old, malfunctioning cellular machinery and replace it with new, efficient equipment. And spinach is the infrastructure, the blood flow that delivers everything to the site, the folate that powers the genetic switches, the magnesium that strengthens the final product. Remove any one of these and the system works less effectively. Provide all 5, and you have given your sleeping brain every major tool it needs to do its job. And there are specific synergies between these foods that amplify their individual effects. The DHA from salmon and the vitamin E from spinach work together to protect cell membranes, DHA providing the structural fat in vitamin E preventing its oxidation. The melatonin from tart cherries and the magnesium from spinach both promote deep sleep through different mechanisms, creating a more robust effect than either alone. The choline from eggs and the folate from spinach are both essential for the methylation cycle, each one required for the other to function properly. The nitric oxide from spinach increases blood flow, which enhances the delivery of DHA, astaxanthin, choline, and every other brain-active compound from the other foods. This is not a random list. It is an integrated system, and the research supporting each individual component is strong enough on its own that combining them into a single dietary pattern gives you a genuinely powerful approach to supporting brain health during sleep. I want to spend a few minutes on timing because when you eat these foods matters almost as much as which foods you eat. Your brain's nighttime repair processes follow a predictable schedule that aligns with your sleep architecture, and delivering the right nutrients at the right time can significantly enhance their effectiveness. Deep, slow-wave sleep, the stage where glymphatic cleaning is most active, and where the initial phase of memory consolidation begins, occurs predominantly in the first half of the night. This is the stage where beta amyloid is cleared, where cerebrospinal fluid flows most freely through expanded intercellular channels, and where the physical cleanup of the brain is most intense. REM sleep, where emotional memory processing and creative integration occur, becomes longer and more frequent in the second half of the night. For the nutrients that support deep sleep and early night repair processes, you want them circulating in your blood and available to your brain during that first half of the night. This means consuming them roughly 2 to 4 hours before bed, which typically aligns with dinner time for most people. Salmon consumed at dinner provides DHA and astaxanthin that will be circulating during those critical first deep sleep cycles. Spinach consumed at dinner provides nitrates that will be converted to nitric oxide over the next several hours, enhancing brain blood flow during early sleep. Eggs consumed at dinner provide choline that will be available for acetylcholine synthesis as your brain enters its memory consolidation phases. Tart cherries are best consumed slightly later, about 1 to 2 hours before bed, to align the melatonin peak with your natural sleep onset window. A small glass of tart cherry juice or a portion of dried tart cherries in the late evening, after dinner but before bed, is ideal timing. Walnuts are somewhat more flexible because the conversion of ellagic acid to urolithins by gut bacteria is a slow process that unfolds over many hours. Eating walnuts at dinner or as an evening snack both work well, as the urolithin production will peak during the overnight hours regardless. This is not about being obsessively precise. It is about developing a general pattern of eating these foods in the evening rather than in the morning, so that the nutrients are available when your brain's repair systems are most active. Before I wrap up, I want to address a few common misconceptions that I see circulating online that might prevent you from getting the full benefit of what I have shared today. The first misconception is that brain health is all about antioxidants. You see countless lists of superfoods that are just ranked by their antioxidant capacity, their ORAC score, their polyphenol content. And while antioxidants are important, they are only one piece of the puzzle. If you are loading up on blueberries and green tea, but not providing structural fats for cell membranes, precursors for neurotransmitter synthesis, or support for mitochondrial renewal, you are addressing one dimension of a multidimensional problem. That is why this list includes foods that work through very different mechanisms. Antioxidant protection is necessary but not sufficient. The second misconception is that you can supplement your way to brain health. I am not against supplements in principle, but whole foods provide nutrients in complex matrices alongside cofactors, fiber, and companion compounds that enhance absorption and utilization in ways that isolated supplements cannot replicate. The choline in an egg yolk comes packaged with phospholipids that facilitate its incorporation into cell membranes. The DHA in salmon comes alongside astaxanthin that protects it from oxidation. The folate in spinach comes with magnesium and vitamin K that participate in related biochemical pathways. These synergies exist because these nutrients evolve together in food, and your body is designed to receive them that way. The third misconception is that what you eat does not matter if you have bad genetics. This is one of the most damaging myths in health. Yes, genetics influence your risk for cognitive decline. Some people carry the APOE4 allele, which increases Alzheimer's risk. Some people have MTHFR variants that affect folate metabolism. But genetics are not destiny. They are a predisposition, and nutritional interventions can dramatically modify how genetic risk actually manifests. Epigenetics, the study of how gene expression is modified by environmental factors including diet, has shown conclusively that what you eat can turn risk genes up or down. The folate in spinach, the DHA in salmon, the polyphenols in walnuts, these compounds all have documented epigenetic effects. You cannot change your genes, but you can absolutely change which of your genes are active. The fourth misconception is that brain decline after 60 is inevitable and irreversible. This is flatly wrong. Neuroplasticity, the brain's ability to form new connections and reorganize existing ones, persists throughout life. Neurogenesis, the formation of entirely new neurons, continues in the hippocampus well into old age, though at a reduced rate. The brain is not a machine that simply wears out. It is a living, adaptive organ that responds to the inputs it receives. Give it the right inputs, especially during the critical overnight repair window, and it can maintain and even improve its function far longer than most people believe is possible. Let me give you a clean summary you can refer back to. Food number 1, wild salmon. 2 to 3 servings per week, ideally 1 at dinner. Provides DHA for cell membranes, astaxanthin for antioxidant protection inside the brain, and B12 for myelin maintenance. Food number 2, tart cherries. Daily, 1 to 2 hours before bed. Tart cherry juice concentrate, frozen tart cherries, or dried tart cherries without added sugar. Provides natural melatonin, anthocyanins for neuroinflammation reduction, and tryptophan for additional melatonin production. Food number 3, whole eggs with yolks. 2 to 3 daily, with at least some at dinner. Provides choline for acetylcholine and memory consolidation, lutein for neuroprotection, vitamin D for hippocampal function, and essential amino acids including tryptophan. Food number 4. Walnuts. 1 ounce daily, approximately 14 halves, at dinner or as an evening snack. Provides ellagic acid for urolithin A and mitophagy activation, alpha-linolenic acid for anti-inflammatory effects, and concentrated polyphenols for antioxidant protection. Food number 5. Dark leafy spinach. 2 cups fresh or 1 cup cooked daily with dinner. Provides natural folate for methylation, nitrates for brain blood flow, magnesium for synaptic plasticity and sleep quality, vitamin K for sphingolipid synthesis, and vitamin E for membrane protection. So there it is. 5 foods. 5 distinct mechanisms. 1 integrated system designed to give your sleeping brain everything it needs to clean, repair, and rebuild itself after 60. Your brain is not a passive organ that simply deteriorates with time. It is an active, living system that fights to maintain itself every single night. But it cannot fight with empty hands. What you put on your plate tonight will determine how effectively your brain repairs itself while you sleep. That is not a metaphor. That is biochemistry. I am Dr. Thomas Reed. If this information was valuable to you, share it with someone you care about, because the people who need this most are often the ones who have never heard it. I will see you in the next one.